Hello, everyone. My name is Alisha Millican with NAMA, and it is my pleasure to introduce tonight's speaker, Damon Tighe. Damon Tighe is a biotech educator based in the San Francisco Bay Area with a unique journey into mycology. His career began with work on the first draft of the Human Genome Project, but his passion for fungi emerged from an unexpected moment of necessity while hiking the John Muir Trail. He ran out of food and found himself staring at large mushrooms, wondering, could I eat that? That moment of curiosity sparked a lifelong fascination. Since then, Damon has become a prolific collector for the California Fungal Diversity Survey, contributing over 1,000 sequenced voucher specimens to the project. He has also dedicated himself to science education, helping numerous high schools develop their own programs to sequence local fungi, bringing cutting-edge molecular biology techniques to the next generation of students, and fostering an appreciation for fungal biodiversity in their own communities. Tonight, Damon is presenting for us Bioluminescence and UV Fluorescence in Fungi of California. Thank you so much for being with us here tonight, Damon. I'm going to go ahead and turn the floor over to you.
All right, really excited to take you guys through this presentation on understanding bioluminescence and UV fluorescence in California. We're mainly going to do fungi, but I'm going to throw in a couple other critters just so that you kind of understand the entire landscape of everything that more or less glows at night if you go out there, either with just your eyes or with UV light. There will be a QR code at the end of the deck. So if you guys want the deck, you can just grab it at the end of the talk, or we could send it out to you later, of course. Already did my background, so I'm just going to jump right into some of like the nitty gritty right away. So there's usually kind of confusion when I talk to educators between bioluminescence and UV fluorescence. So I'm going to kind of set the ground for both of those and then show you examples of them. So bioluminescence is when something produces its own light via an enzymatic reaction. So it's going to take some sort of substrate, and as it breaks that down using an enzyme, that energy that's released during that reaction will release a photon of light in a spectrum that we can see. There's probably organisms that are releasing light in spectrums we can't see. I mean, there's obviously, because things also let things go in heat, which is also could be a spectrum. But for the most part, when we think about fungi, this is going to be stuff in kind of like the green spectrum. And there's a variety of mushrooms that do this across the world. And we've got a number of them in North America. California probably has fewer of them than, say, the East Coast, because you guys have all this kind of like semi-tropical vibe going on with a lot of your fungi. And I know when I talk about bioluminescence with people, usually the first thing that comes in most people's minds are fireflies, right? If you ask the general public, like, tell me something that glows on its own, people are like, fireflies. And in California, everyone is super disappointed because they are convinced that we don't have fireflies. We do have them though. We just don't have ones that fly that when they glow. So for us, the larval stage of our fireflies are what glows. And so the kind of colloquial name for them is then the pink glowworm, because this is what you would find if you find one of these things out in the forest. My first experience with these was pretty freaky. I was doing a section of PCT at night just because we were trying to cover ground when it wasn't hot out. And we were hiking through the thermal areas near Lassen, which is a volcanic area. So the ground is literally steaming and things like that. And we could see these lights like blinking in the distance, and we are way far away from the road. And so we're like, who's there? Who's there? It's not answering back. And it kept moving. My friend's like freaking out. And I'm like, I'm just gonna go try and figure out what it is. Sneak up on it, and it's this. So bioluminescence, kind of the hallmark image for most people is, a firefly. So California does have those, they just don't fly around. And then just throw it out there, they're not really flies. This is when common names, right, sometimes cause problems, not just in fungal taxonomy, but in other organisms. So these are actually beetles. So this is a beetle larva that you see here, and the flying ones on the East Coast are beetles. The cool thing about bioluminescence compared to UV fluorescence, which isn't as well studied, is that based upon the spectrum of light that you see, you could actually take a pretty good guess at what organism is producing the light. So if I see something like in the bluish spectrum, these tend to be more marine organisms or organisms you would find in aqueous habitats. So like ostracods, the Cyprinidae, those ones will kind of pump out kind of blue light in water. Shrimp produce kind of blue light, some of these shrimp that glow. Or a lot of the experience I have in California are these things that are zooplankton, some of the bacteria and some of the other protist groups that will produce blue light in water. And the reason you probably see blue light in water is if anybody here is a diver, you know red light doesn't penetrate very far through water. And so if you're pumping out blue light, you're going to get a lot more bang for your buck. It's going to go a much bigger distance. And then when you get to the terrestrial places, fungi, for the most part, everybody that we know so far that produces bioluminescence as a fungi pumps it out in a really discrete range. It's right in the spectrum of green light. And so, we don't have any red glowing mushrooms out there, no purples, no blues. Everybody's more or less pumping out the same wavelength, this green spectrum. And then not too far away from the mushrooms, we see things like beetles that also pump out some green light, but it's usually more kind of shifted towards the green yellow spectrum. So that's going to be your fireflies. It's going to be your click beetles and your railroad worms. Although there's some weird railroad worms out there in the tropics that will pump out like red light. But the cool thing with the bioluminescence is if you can see the light in the distance, you can take a pretty good educated guess on what type of organism you might go find if you crawl up on it. So in iNaturalist, when I first started observing this stuff in California, I made a project for it. And so now this is iNaturalist, of course, is global. And so this project is global. You can go there and really quickly figure out what organisms around you are actually bioluminescent and then go look for them. And you can see the total number of observations there is really low. And it's not because people aren't seeing stuff glow. It's usually it takes a while for people to be able to figure out how to take good photographs of them and then get them onto iNaturalist. So we'll cover that kind of at the end of bioluminescence, how to get a good photograph. Because a lot of the mushrooms in North America that I think glow, we just don't have images of them because it does take somebody with a little bit of photography skill to sit down, do a long exposure to then say, here's evidence that this is glowing. A lot of our mycena glow, and not many of them have good photographs yet. And so folks like Alan Rockefeller do a lot of these. I try and do a lot, but it'd be great to get more of you out there photographing mycena for long stretches of time at night. One of my first experiences with bioluminescence in California was this really weird one in an urban kind of estuary where I live, which was the water in this urban estuary, which most people, even if you just tell them you're touching the water, they're like, you might catch a disease. So we have an urban lake in Oakland called Lake Merritt. And at one time of the year, basically September through October, on a new moon, the water will glow bright, bright blue. So when I first started like seeing this, I thought, you know what? We'll sweep this out, do some microscopy. We'll know who this is. I send this out to all the experts and they're like, oh yeah, the list for things that are possibly causing this blue is like super, super long. We could maybe get you down to a genus as best. And this recurs all the time. So if you're in any of these areas where you have like a marine estuary, you have brackish water, your chance of seeing bioluminescence is relatively high. It's probably occurring at some point of the year there. You just need to figure out when it is. And it tends to be whenever you have big algal blooms. And it tends to be right on the back end of the algal blooms because a lot of these things are eating those things that are blooming and then using that energy to be able to run bioluminescence. Why do these marine critters do this? A really interesting strategy is if you're a really small critter in the zooplankton, you have little small things eating you, right? And what's going to prevent those little small things from eating you? Bigger things eating them. And so if something swims through you and all of you and your buddies glow all of a sudden, it lights up the predator. And all of a sudden, that little small predator fish is visible to other fish, which then can come and eat it. And so this is an interesting kind of hypothesis of why some of this bioluminescence is taking place, at least in these marine environments. We also have things like brittle stars in California that will glow under their own direction. Fireworms as well. So a lot of marine organisms utilize bioluminescence to scare other organisms or to light up predators. So fireworms and starfish will use bioluminescence basically to get things to stop attacking them. Because if they can hit them with light, it scares the predator away. So a lot of these are kind of defenses against crabs and fish. Skip that one. Let's get into the fungi. So in kind of Myceniaceae, there's a lot of things that have UV fluorescence. One of the coolest ones, I think, are the dripping bonnets. So in the West Coast, we have Rhodomyces rhoridus, and I think you see this across most of North America if you get to kind of like places that stay kind of humid and wet for a long duration. And these are an interesting group because they produce these like little slime droplets right at the base of the stipe. And that is there probably as a defense against arthropods. They're trying to climb the stipe and then get to the gills, right, to eat the goodies. And so the mushroom is basically using this to protect the gills so that it can get spores out in the world. And every time I've photographed this species, the only place it seems to glow is right around the droplet of slime. And so arthropods can sometimes be attracted to light. And so I think what might be going on here is that this mushroom is using that little bit of production of light to then bring things like springtails and stuff like that into the slime so that way they can't get at the gills. Some of these are really, really faint. So with the naked eye, your chance of like seeing it in the forest is relatively low. But if you take it home and you do a long exposure, like a couple minutes, like in a dark room, that's when you can start to see the glow on these. And unlike some of the tropical ones, a lot of our Mycena, at least in North America, don't seem to have the actual fruiting body glow. Other portions of the fungal structure are glowing. So either things like the stem, the stipe, or the mycelium itself, which brings up a lot of questions about why would you want to do this? So also in Rhodomyces, you can see that the mycelium occasionally glows. So if you look at the substrate they're attached to, like this leaf fragment here, you'll also see that it glows if you do a long exposure on it. The bleeding fairy helmet, which we've got distributed all across North America, has really, really bright UV, or not UV fluorescence, but bioluminescence, but it's just the mycelium. And so if you take home a log with fairy helmet on it, bleeding fairy helmet on it, and you just cut it with a knife and then take a photograph of it, you'll realize that everywhere the mycelium's running in the wood glows. And so why are they doing this? Who knows? Some hypotheses are they're just trying to get rid of excess energy, and this just happens to be a way to do it. Is, instead of giving off heat, you're just giving it off as a photon of light. It might be that maybe this is a strategy to maybe move, particles of the fruiting, not the fruiting body, but the mycelium around by attracting insects that are already burrowing through the wood. They see a glow, they go over there, they get maybe dusted with a conidia or maybe even just a broken piece of mycelium, and they take it someplace else in the wood. A lot of like big questions still to be answered on this. Purple edged bonnet, Mycena purpurefusca that we see at least out on the West Coast a lot of times on conifer cones, specifically like Monterey pine, they seem to like, is very similar. The fruiting body does not glow, but the mycelium glows. So if you basically cut wood that they're on or you cut a cone that they're on, you'll realize that that mycelium is glowing. So these photographs are by Alan. And Alan does a lot around bioluminescence, especially Mycena, like in Central America. And then quite a few of his images are in this deck, just because he's taking the time to do these long exposures of ones in California. The bulbous bonnet also glows really brightly. This one is actually a little bit brighter than the rest of the ones that we see in California. So this one, if you're walking in an area where this one is fruiting, a lot of time the needle duff, you can actually see a faint glow. But the caveat there is you need to just go for walks without a light. So walk relatively like well-known trails to yourself without a light, because it takes, 10 minutes for your eyes to adjust. And then once they adjust, you'll be able to start to pick up this green with your eye. The crazy thing though is from a photography point of view, you'll be like, oh, that is super bright. And then you'll put your camera on it, and your cameras a lot of times seem to really struggle with this spectrum of light that fungi produce. And so the exposure time usually ends up being a lot longer than one would anticipate if you're used to already shooting kind of long exposures. One of the coolest ones for bioluminescence, especially the mycelium, is the armillarias. And so we've got 4 or 5 on the West Coast, and I think we've got documentation now that at least 3 of them have really strong bioluminescence. And so this is one in culture, I think that Hart picked up and then Alan photographed once it was in culture. And what you see in the mycelium here is a couple interesting things. You see this kind of white fluffy mycelium, which is kind of what most cultures tend to look like of Basidios mycetes, right? But then you see the armillaria starting to weaponize. You see those kind of browner, like thinner runners, and that's where they're starting to aggregate melanin. So it'll use melanin as a physical structure to then basically have like a weaponized piece of mycelium that can jam into plants. And all of that glows, the fluffy stuff and the weaponized stuff. And so my first experience with these was as somebody growing up in like rural California, and a friend was removing a stump at his house. And like most home projects, they take longer than you expect. So you start in the afternoon, you're like, I'll be done by dinner time. And then like it's 2 hours after sunset and you're still working on this damn stump. And the stump all of a sudden is glowing. And so what we realized was it was a tree that had been killed by armillaria. There was still mycelium inside of it. And so we were seeing just the glow from it there. And I get reports occasionally from the Pacific Northwest of people taking out trees or after a storm when a tree has its bark get peeled off that's been killed by armillaria. Well, we'll just have a glowing, basically totem pole of armillaria mycelium, which is pretty wild. This, if anybody out here is like an installation artist, this one would be a great installation project to make a giant auger, grow out armillaria because it already looks like something nebulous, looks like some sort of like dark, deathy, like heart growing out of there. And then it would be able to glow at night enough that people could see it. So it would be a really cool installation project if somebody wants to go about that. In California, our brightest glowing mushroom, the one that you can actually walk through the forest and see at a distance, is Omphalotus. And just like the East Coast, Omphalotus tends to be some of your brighter mushroom species too, and they're very large, right? So they're like they are the hallmark of like bioluminescent mushrooms in North America. Although ours pale in comparison to the Australian one. So if you ever get a chance to go to Australia, time it just right to see theirs because it is unbelievably bright. But Omphalotus olivescens is the one that we see in California. And this is a mushroom of just floristic California. It basically runs into Oregon a little bit, runs through California, and then gets into Baja, Mexico a little bit. So it's really kind of restricted in range. And this is why, for a long time, we were trying to get this to be the state mushroom, but it lost out to chanterelles. So now we're just like every other state with a chanterelle as the state mushroom, where how many of the people have a glowing mushroom that you could also dye stuff with? They get really big. They make a wonderful lavender dye. So I was at a high school and some girls that we were doing a foray with to do sequencing had found like a really big population of them. And they asked the teacher, and this can only happen in like small schools. They're like, Mr. Gady, could we not go to physics and make a mushroom dye from these Omphalotus we found? And I was like, that's never gonna fly. And he's like, yeah, let's do that. So these girls happen to have a spool of yarn in their bag. I don't know. Crush it up with a little bit of vinegar, dumped it in there, and you get this wonderful lavender color. If you add a little bit of iron to it, you get a really, really nice green, like chartreuse color. So it's a really cool mushroom all around. Um, One of the ones that's, at least for me, always been the biggest disappointment in, oops, hold on one second. I'm missing Alisha. Yeah, give me 1 second, folks. Is everybody here still? I think we just lost Alisha. I just want to make sure that everything's going okay. Can somebody just in the chat say that you can see what's going on and I'll continue? All good? All right. I'll wait for Alisha to kind of jump back in. Okay. So let's go back, to the bitter oyster. And let me just let her know that we're still running in the background. All right. So bitter oyster on the West Coast, for the most part, does not glow. My only chance seeing it is when I went to Indiana for work once, and I was blown away. It's a really, really bright mushroom, right? It's one of these ones you can see easily by walking through the forest at night. But from a sequencing point of view, we appear to have this in California, but very, very few of ours seem to glow. So we don't know why that is. Like, what is the biological importance for this mushroom to have bioluminescence? And why do we seem to see it shift when you get to the West Coast? Like, why is it not producing the bioluminescence? My gut feeling is there's a vector that it's using out east that maybe we don't have out west. And so there's not this kind of evolutionary pressure in order to make it glow as much out here in California. So for years, we've been looking for one that glows. The only documentation we have is, I think, a single observation that Alan got maybe a year or two ago, where it seems to have bioluminescence out here, but very, 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 very faint. So what did you guys notice about all these ones that glow? Well, one of the weird things about them is that all of them are saprophytic. Like, we're not seeing any mycorrhizal mushrooms, right, that glow. It's all these ones that are breaking down some sort of, lignin and cellulose structure. So if we think about the kind of phylogenetic tree of where these occur, they're all just kind of like in generally one branch. And that's more or less going to run from like Omphalotaceae over to your kind of Mycenas. And it's just this little section of the tree that has things that are producing bioluminescence. But this is from Brian and et al.'s paper from a couple of years ago. And this actually is a really, really nice tree to have because if you're going to go out and try to document stuff in your area that might have bioluminescence that nobody has captured yet and seen, this is your roadmap. I would start with the Mycenas just because it seems like there's a lot of them that glow. But then I would kind of, thumb through some of these other ones that have things that glow and maybe just do long exposures to see if you can pick up stuff that we're not seeing normally with what's there. So why is this? Oops, sorry, guys. I'm trying to get Alisha back in. So most of these are running the bioluminescence on what's known as the caffeic acid cycle. And the caffeic acid cycle, some of the components you need that are running into this mainly come through the degradation of kind of like woody substrates. And so that's why we're predominantly seeing these saprotrophic fungi that are producing the glow. And so this caffeic cycle is kind of cool because you can manipulate it. If you put something in culture and then dump more caffeic acid into the culture, you can kind of drive the bioluminescence a little bit stronger. And then because this is this cycling process, it basically will regenerate itself. And so you can keep a culture that glows relatively well over a good period of time. Now, why are these doing this? Well, there's been a lot of hypotheses out there and testing, as far as papers go, there's not a ton of papers out there at this point. But there was one that came out not too long ago that's really interesting because what they were noticing was that some of these bioluminescent mushrooms have a circadian rhythm to them. And so if you just have the mushroom isolated and you're watching it, it's kind of coming up and peaking how much it glows, like on a rhythm. And that rhythm is the bioluminescence is strongest more or less in the middle of the night, right? And from like, if you're trying to get bang for buck, right, of like running this system in your biology, that's probably where you want to do it, right? You don't want interfering light from sunrise, sunset, or daylight in there. But then the other question is like, why would you do this? This is a lot of energy potentially that's being invested in the system to produce light. And so one of the going hypotheses is that it is a way to find yourself a vector. So you can take these night flying insects, emit at a wavelength that attracts them. They come over, check out what's going on, get dusted in spores, and then fly away with your spores and then take them some other place. And so this paper, what they were seeing was that what seems to be the dominant things that are probably moving stuff around are probably Diptera, so probably flies and beetles, which wouldn't be too surprising thinking about even those ones that might be burrowing through the wood are probably going to be beetles. And then flies are they're a great vector for lots of things. I know everyone hates flies because they get on your food and stuff, but I mean, they're one of the main pollinators in the world too that never get the daylight. They're extremely important in driving a lot of vectoring of pollen and probably spores in the world. A really interesting kind of mashup of plant meets fungi is that in the past 2 years, there was a company that released a direct-to-consumer GMO where you can basically buy a petunia where they move the whole entire bioluminescent pathway out of Neonothopanis namibii, which is a South American mushroom that's got a nice strong bioluminescence, into this petunia. And being somebody that works in biotech, I mean, I've seen a lot of like glowing plants, like glowing tobacco that use luciferase, and they all suck. Like you'd see them, and you're like, it's not glowing. And people would be like, oh, you got to go in the dark room and let your eyes adjust. You're like, you're in there for like 10 minutes. You're like looking at the tobacco plant. I still don't see it. And they're like, do you have a camera? If you take a long exposure, you could see it. Where this is my plant from at home. I can literally take it from outside, just walk into a remotely dark closet, and I can see the blooms glowing, which is pretty wild. So this is going to be something I think you'll see more sort of like weird consumer products coming out of where we take bioluminescence and try to stick it in plants and other things. Photographing these bioluminescent fungi can be very, very tricky. The biggest thing is you want to get a way that your camera isn't being touched by your hands at all while it does the photograph. because even just your heartbeat coming through your hand and touching your camera will shake your camera when you're doing a long exposure and things won't come out clear. So the best thing is like use a tripod or use something to rest your camera on, and then set up how to do a long exposure. So for a lot of things, when I'm trying to like determine whether or not is this worth my time to try and do a longer exposure, what I'll do on my camera is I'll do what I call a test shot. I'll basically drive the ISO ASA of my camera up as high as possible, so like 6400 or higher. I'll do a shutter speed of like 30 seconds and then the lowest f-stop I have. And I'll do a quick shot. And if I see a little bit of glow there, then I will be like, okay, I'm going to invest time now to try and get a good photograph by decreasing the ISO ASA, which is going to give you better resolution, better color images, things like that. And then extending the time out until I can kind of get that glow. A lot of the things that you saw as far as the mycena that I photographed, you're looking at like a 5 to 10 minute exposure at a low ISO, but I can usually see them right away with a 30-second sort of like test shot with my highest ISO. So that's your best bet for like learning to photograph them for the first time. And probably the best way to do this is go after something you already know that's glowing and then kind of get your process dialed in to do the photography. And if you ever need help with that, feel free to reach out to me. I'm happy to troubleshoot directly with anybody. So let's do a crossover organism before we move into UV fluorescence. In California, we've got millipedes that actually have bioluminescence. So they will glow on their own. There's a couple photographs in INAT of like people holding these in their hands, and like you can actually see the texture of the person's hands because they pump out so much light. But they also do UV fluorescence. So they are basically the thing that glows in both ways. To find these in California, most of them occur in the southern Sierra Nevada mountains. And then there's a really disjunct population in San Luis Obispo County on the coast. I saw these in slow, but I didn't know they had bioluminescence. So all I have is this UV fluorescence shot. So what is UV fluorescence? UV fluorescence is very distinctly different than bioluminescence. With UV fluorescence, you're going to basically hit the organism with high-powered energy light in the UV spectrum. And what's going to happen is something inside the organism will then release light at a different, lower intensity wavelength that then is in the spectrum that we can see. And so for you chemistry nerds, we can go a little deeper in. And for those of you that didn't like chemistry, I apologize for the PTSD. We're going to talk about electron orbitals. So you've got electrons, right, that are floating around, your center of your atoms, and they're kind of in these shields, and we give them like orbital names. And what can happen is you can transition from one low state, which they're usually at, to a higher state. And these electrons do not like to be there. That UV light, when it comes in and hits the compound inside your mushroom, what's happening is it's actually pushing those electrons up to the next orbital. And the electrons do not want to be there. And so what they will do is when they fall back down to their ground state, they'll release the energy from that higher state as a visible spectrum photon. And that's what our eye is going to see. And because you can have all different sorts of electron orbitals depending upon your compounds, this means we're going to get a broad shift of spectrums that we'll see. It's not going to be just green for mushrooms. It's going to be all over the place because we're illuminating a big variety of compounds. A lot of the compounds that will do this tend to be things with a lot of double bonds and aromatic rings. So one of the easiest things that you could like put a UV light on in your home is if you have turmeric. Turmeric has this compound in it called curcumin, and it is crazy bright. I used to be an apartment manager, and I had a tenant who cooked a lot with turmeric. And when he moved out, I just thought, oh, it might be probably not something you ever want to do. Don't do this in a hotel. Take your UV light and go into a room and shine it around. So I did this in his kitchen, and you can't see it with your visible eye and invisible light. But I mean, everything. It was like a Jackson Pollock painting of like bright yellow all over the place, because wherever just a little microgram of curcumin ended up on the wall, it would just glow. If you think about other things that fluoresce, like people a lot of times in the biological world are fascinated by the fact that scorpions fluoresce really, really strongly. And this is usually due to two compounds you see in their exoskeleton, which is beta-carboline and 7-hydroxymethylcoumarin. And once again, these things have big aromatic rings with lots of double bonds. That's going to give you that more or less electron orbital structure that will allow for that UV fluorescence. So for a lot of mushrooms, I think what we're looking at tends to be alkaloids and then fats that are doing the fluorescence. So just since we were just talking about millipedes, I'm just going to highlight another millipede that we get in California, which is Zostria dissecta. And this is probably the most common organism you can find on a hike in our wet winter months when you're looking for mushrooms in California. And it glows so strongly, you can see this at about 50 feet with a UV torch. Really, really bright. The nice thing is, from a fungal point of view, is these guys get a really, really cool fungal infection. They get this thing kind of analogous to like fly death fungi. And we don't really see this very commonly on the West Coast. And I think it's mainly because people just don't know how to look for this. But it's this fungus that takes over the nervous system. These millipedes will crawl up high, they'll die, and then the fungus erupts out beneath every single plate and then starts shooting spores out. We don't have a paper on this yet, but I wouldn't be too surprised if it is also like fly death fungus in that it is timed. So fly death fungus kills the fly right at sunset. So it'll infect the fly gets summit disease, so it'll crawl up really high. It will then stick its little, proboscis out. Fungus grows off of it, holds on to the substrate, and then right at sundown, it then kills the fly. And then what happens is over the course of the next couple hours, the fungus will emerge and then start shooting spores. And the reason you might want to do this is if you look at this, which is very similar to fly death fungus from like a macromorphology point of view, these spores aren't pigmented. So you're shooting spores when there is the least amount of UV light around, so you don't have to worry about your spores being damaged. And in the case of the flies, this is when all your flies are probably hanging out, not moving. So if you climbed up high, you're raining spores on them. And the exact same thing here. This organism is most out and about at night. And so if you're able to kill it at night and have it shoot spores at night, you then can rain it down on the rest of the population of millipedes. So the easiest way to find this one, at least in California, is go out in our winter, find a big population of these, and then just start looking around for ones that are high off the ground with your UV torch. And if you go over there, like 95% of the time, they're not moving, and then you'll see the eruption of the anthrophagia fungus from beneath it. This one in California is still undescribed. We've got sequences from it from the California Fundus Project, but still undescribed. And if you would like those, they are in the Hayward Fungarium if anybody wants to work on that. UV fluorescence, we do have an INAP project around that. So any fluorescence that you see, you can put it in this project, which has been, from like a community science point of view, this has been one of the funnest projects to be a part of, because even academics weren't going out there with UV lights for very long ago. And so there's still this huge, broad world of UV fluorescence that people are discovering. And a lot of this is done through people that are in not academia. They got a cheap, LED light and are just walking around finding stuff. And we're learning all kinds of really interesting biology around this. And some of these are turning into really important biological tools. So like one of the ones that's very interesting is owls now. A common practice is to sex the owl and get an idea of the owl age based upon its UV fluorescence. So owls' UV fluorescence is stronger in females, and then it declines in age. So based upon the fluorescence, you can get a really quick idea of like, is that a female, and approximately what age is it? So I just did this talk for a group in California recently, and like the Audubon Club there that does tagging was like, oh yeah, we do this. We've been doing this for like at least half a decade now. So some of this UV is becoming real protocols for people, which is really, really cool. When most people think about UV fluorescence, though, they think about scorpions. Every scorpion, from what I can tell in the world, fluoresces due to those compounds we talked about before. We've got a variety of them in California. But one thing to think about with this, and this is something that also might carry through to some of the fungi not doing fluorescence at certain portions of their life, is you have to build up those compounds that are doing the fluorescence. So this is a scorpion I saw last year that had a bunch of babies on the back. And what you notice really quickly is none of the babies are doing UV fluorescence. And it's because they're not old enough to have those compounds aggregate into their exoskeleton and glow. So just mama glows and not the babies. Possums and a lot of mammals have UV fluorescence, and they can be very, very striking. The mammalian kind of academic world is like going down a rabbit hole on this because in some of these groups, we think that they might actually allow organisms to recognize each other because they'll have different kind of UV fluorescence on the face. For most possums that I've seen, they're all just super hot pink, but portions of the face will be blue, and then portions of the tail will be blue as well. Rats also fluoresce really, really blue. In fact, if you're trying to find a rat's like in your house or in your yard at night, the easiest thing is to throw a UV light out, and I mean, they glow crazy bright. Let's get into the fungi. Bird's nest fungi have a lot of UV fluorescence, but only when they're really, really young. And so if you take a bird's nest fungus and you do a cross section of it and you throw a UV torch on it, the petioles are usually really, really bright. Have no clue why this is. And then also that kind of like hydrogel like material that's in there with the petioles will also usually fluoresce very strongly. But as they mature, this fluorescence goes away. So in the back of my mind, I think this is probably some sort of lipid that the fungus is maybe using as an energy source or is getting transformed in some way that once they're mature, it's used it up or it's just degraded. Gymnopolis are probably one of the generally brightest groups of fungi that you'll see with UV fluorescence. In fact, I would say some Gymnopolis I can't find without a UV torch. And this is the one that I can't find without a UV torch. This is punctifolius, which is one that loves rotten coniferous logs, mainly Pacific Northwest. And I mean, it hides inside the log. Like you've got to usually dig inside the log to find them. But if you go out with the UV torch at night, just that little edge of a mushroom that might be poking out, you'll hit it from 20, 30 feet away and you'll see that bright green and then be able to come over and find this really, really bright gem there. Powder caps are another group that have some really, really cool UV fluorescence. They actually kind of feel like you're in like a fairy tale land because of the way they give off their glow. It's predominantly in the gills and it's kind of this shifted blue green. Really, really beautiful under a UV light. A really interesting one, striking one that's super common that basically anybody could find because it's freaking everywhere, right? It's on every continent now, is Schizophilum commune. It has bright blue gills. And we don't see bright blue gill edges on many things. And so when I first saw this, I was like, what the hell is going on here? Like, why is that? So I took it underneath the microscope and tried to make like a DIY UV scope at home, which basically means you put your camera in front of your microscope, so you're not putting your eyeballs in there, take a UV light and shine it at about a 90-degree angle. And this is how you're going to get the best UV fluorescence if you're doing a DIY scope at home. And what I could find is it seems like some of the cystidia have some sort of inclusion in them that have this blue fluorescence. I have no clue what this is at this point. Is this a fat? Is this an alkaloid? I don't know. But this is definitely one, if you're just kind of breaking into UV fluorescence, you can for sure find this mushroom, hit it with a UV light. You want to find a really fresh one in order to get the really blue glow, but it is stunning. Another really stunning one is Entoloma sebcarneum. And this is one that I think me and Alan have documented a couple times with UV fluorescence. And I mean, it looks like AI because it's just like so crazy looking under UV light. And I always thought this was gutation, just based upon where the fluorescence was. And when I found these, they're always like super moist and wet. And then at Soma Camp this year, this really, really cool, like weekend-long thing that the Sonoma Ecological Association runs, we had this, one of my favorite young mycologists, Caitlin. I think she might be pushing middle school at this point, but I think she knows every mushroom in Mushrooms of the Redwood Coast and like can just talk circles around every adult. And she's super cute and it's like just completely disarming for everybody. She found this and knew about its fluorescence and like brought it back and was like, we got to look at this. And then David Healy at Sonoma Camp got this underneath the microscope. And what they figured out was that it's the spores. It's not gutation that's glowing. So there's some sort of inclusion in the spores that actually is causing the fluorescence. And so we have no clue what that is right now, but like all the spores that don't have an obvious inclusion didn't glow, and all the ones that do, boom, bright green under UV fluorescence. So super cool. Hypholoma. If you're looking for a global species or global genus that has strong UV fluorescence, Hypholoma is where to go. The sulfur tuft, at least on the West Coast, is super, super bright. It's one of these ones that you can see from 20, 30 feet away with a UV torch. And it's always the one, if you're leading a UV walk, you always want to have Hypholoma somewhere in the walk because everybody will just scream because it's so bright. Really, really, really cool mushroom. And what I liked about Hypholoma and kind of looking at multiple species is that in this process of using that INAT project, we kind of developed a hypothesis that maybe all Hypholoma glow. And with that iNaturalist project, it's really easy to test that because I can just go into the UV project, I can type in Hypholoma, and I can see all the Hypholoma that people have documented that have UV fluorescence. And then I can take it against our known species list for Hypholoma and be like, where don't we have data for this? And then I can just go on INAT and bug whoever sees that mushroom a lot in that area and be like, hey, can you throw a UV torch on it? And then we can get that data. So a really, really cool way to use that project is to ask questions like this, like, does this entire genus have UV fluorescence? And in the process, you get to meet cool people from across the world that are doing nerdy stuff that you're doing. Cortinarius. A lot of quartz have like mild UV fluorescence, but the section Leproscybe is jaw-dropping because Leproscybe normal is not jaw-dropping. In fact, you'll probably just walk by them in the forest and not even realize that they are there. But under UV light, all of the tissue glows, all in this same kind of like radiant green gold color to the point where you can't like look away from it. And Leproscybe, unless you're really good with your keys, is sort of hard to key out sometimes. And I don't know how many of you guys are like quartz experts out here, but I'm certainly not. So I love being able to throw a UV torch on and be like, okay, I know what section I'm probably in right now. Galerina is another cool group when it comes to UV fluorescence. A lot of the cystidia on Galerina appear to fluoresce. And so most species of Galerina that we've looked at least in the California Fungus Project, where we mandated that people had to throw a UV torch on everything we collected more or less, has really, really cool fluorescence. Usually it's just the gill edges, but a couple of them, like Galerina badiapes here, will have fluorescence that kind of tails up into the gills. Some of them will be a uniform green across all surfaces, but Galerina in general have really, really nice UV fluorescence. So definitely one worth throwing your UV torch on. Hygrophorus is another group that has a really cool fluorescence because it's got this kind of like fairy tale blue glow to it. So pretty much every Hygrophorus I've ever looked at will have this very similar blue hue to it. And then a good majority of them, if they're getting damaged by something, and then I think this is a bacterial infection that might be causing it, they'll have this really cool pink color just where there's damage. And so what I've noticed is with UV fluorescence, you can tell where damage is on a lot of fruiting bodies because it will shift the fluorescence drastically. And in fact, on some mushrooms, you can do this on your own if you're already KOHing your mushrooms. Where you drop your KOH line, hit that with the UV, and sometimes you'll see that shift the UV fluorescence. Sometimes it'll kill the fluorescence. On quite a few of the Cortinarius, it'll cause it all of a sudden to fluoresce when it doesn't usually fluoresce. And right now, I do not know the chemistry of it, and I haven't met anybody that knows the chemistry of it. If you have a chemistry background and you know a little bit more than this, I'd love to get you on the phone and learn from you. Phycomyces, that thing we all try to walk past and not touch. Some of them have some crazy fluorescence, but it seems to be not necessarily where the spore production is taking place. It looks like it's part of the guttation around kind of like where it's wrapping up the turd usually glows really, really brightly. So don't just walk past those Phycomyces. Go ahead and throw a UV torch on them. Ganoderma, there's been a lot of speculation by people that are into shell fungi that maybe at some point UV might be a tool for understanding which Ganoderma you're looking at, because it seems that some of them have some really distinct colors they do or do not get to depending upon the species. I'm still waiting for somebody to write that up in a way that I completely 100% believe it. But what I do believe is if you see a Ganoderma, it is worth throwing a UV torch on it at night, mainly because on a lot of them, at least things like Artis conch, Appellatum, brownie eye kind of group, you'll see this kind of shift from like a blue purple out to a pink on different parts of the mushroom. And if it's one of the ones that's producing a nice lacquer to it, a lot of the lacquer will fluoresce really brightly green. So they're really, really beautiful mushrooms under a UV light. Probably my favorite group, though, of mushrooms to throw UV light on is Diconica, because Diconica are not the easiest mushroom to always field ID, right? Like they fall right into that LBM group really quickly. And then there's a couple real jerks in there that don't even want to pretend to be an LBM because they'll almost like pretend to shelf out, like Diconica horizontalis, like this one right here. But the wild thing about Diconica is every single Diconica I've looked at so far under a UV light has just really nice red fluorescence on the gills. And this is something we really don't see on many other genera. The only other genera that we tend to see this on are like lichen folia and things like that, which you could easily separate from Diconica visually. So this is a really nice tool potentially to say, what I'm looking at here is Diconica and not one of the other LBMs. A lot of us have been playing with UV now for about, I think my first foray into this was maybe 10 years ago at the UC Santa Cruz Fungus Fair, where somebody let me in there at night with a UV light so I could just go look at all their samples. And I was just like, oh my God, this is incredible. Um, but finally, I'm actually seeing UV fluorescence being used in a key to define a new species. So Gliophorus calunis, which just published recently, this is actually in part of the key for it that helps you differentiate this against another Gliophorus in the area that can look similar. Is this kind of blue fluorescence that you'll pick up in the gills. And like on this one right here, you can see where the gills have damage on them, right? They're producing that kind of like pinkish peach color again. So this is an emerging tool that I think we will see get more and more use as time goes on. Also, I know that a lot of us struggle with russulas to the point where you see a russula in the field and you're like, I don't even want to look at it because I can't put a name on it. Go out with a UV light and you'll find that you love russulas again because all of them fluoresce crazy bright. Most all of them have bright UV fluorescence on the gills that's using this really cool green spectrum. And then a lot of them will get a flush on the cap that is blue. And I'm not sure what that flush is, but I'm not 100% convinced that it's actually the fruiting body producing it because I've been able to take this off of it, put it on an auger plate and grow out the flush. So I think it's actually maybe a bacterial component, maybe something like Pseudomonas, which is a known group of bacteria that will fluoresce that's maybe causing the flush on some of these russulas. Lichens, of course, also have fluorescence, especially if you have lichens in a zone that get lots of UV exposure. A lot of them are producing compounds to capture UV light so that they don't damage the fungal component or the photosymbiont. And so most of the ones that are getting blasted by light out on rocks and stuff like that will have some degree of fluorescence to them. But then a lot of other lichens in deep forests will also have fluorescence. And some of that is caused by the photosymbiont. So chlorophyll A produces a red fluorescence. And so in a lot of these fungi, we can see a red fluorescence in them. And if you really want to have an experience, no matter where you're at, go out into a forest after you've had a really, really long dry spell and you've had your first good rain of the year. And what happens is all of these lichens that have been building up these acids and alkaloids during that dry period will shed them on that first good rain. And I mean, it's basically like going out into the forest and not needing to ingest a mushroom to have a mushroom experience because everything on the trees is glowing. It's really, really incredible. It feels like walking through an Avatar movie a lot of times, going out on that first rain. Slime molds have a degree of fluorescence. It's usually the plasmodial state of the slime mold will have it. And then as soon as they start to make the mature fruiting body, the fluorescence falls off. Which is really interesting. I'm not really sure what's going on there. A lot of mollusks will have fluorescence. And so if like in California, if you're in a coastal area, if you find things like nudibranchs, a lot of your nudibranchs will have this blue fluorescence under UV light. And if you get to your terrestrial snails that are eating your mushrooms, most of them will also have really, really beautiful blue fluorescence on the actual tissue. And then sometimes you'll get fluorescence on the shell if the shell has very, very kind of minute features to it. However, one of our non-native snails that we find in North America that came from Europe has a completely different fluorescence to it. It'll be bright green. So if you have a garden and you have a difficulty dealing with garden snails, get a UV light out and go out at night, and it's super easy to find them because they're extremely bright. It'll also help you find things like tomato hornworms. A lot of the caterpillar stages that blend in with plants are very bright under a UV light. Um, Chitons, you guys don't have to worry about that. Spiders are an interesting one. A lot of spiders fluoresce, and this might be kind of going two different directions. Sometimes they're hiding in flowers and kind of pretending to basically blend into the fluorescence of the flower. And then some spiders, if you're walking out at night with a UV light, what you'll realize is you'll find them hanging out on the tip of a plant and basically pretending to be a flower at night. And so insects flying by can pick up a little bit of that fluorescence or a little bit of that spectrum and then fly towards them and then get captured. So a lot of the arthropods will fluoresce. So things like wind scorpions will fluoresce really, really brightly. A lot of your spiders, like I said before, will fluoresce, especially around the joints. And then any of the web spinning spiders will usually do that same fluorescence that these crab spiders are doing. They'll hang out in the middle of the web and then basically have this really strong UV fluorescence, which I think is kind of tricking insects to come in thinking that it might be a plant. And then they get trapped in the web and eaten. You see this with beetles that also hang out in flowers. They will also have strong UV fluorescence. My guess here is they're not trying to be a predator. They're trying to have the same fluorescence as a plant to then not be eaten by a bird. So they blend into that spectrum. If you can get access to the marine environment, anything that's a cnidarian, pretty much sea anemones and jellyfish will glow like crazy. In fact, one of these compounds that comes out of all of these cnidarians is something that I use a lot in biotech, something called the green fluorescent protein that was discovered in a jellyfish. It's all on the West Coast, but it was discovered up by Seattle. And this is a small protein that, from a molecular biology point of view, I can put this next to a gene in any cell. And when my gene of interest gets turned on, this protein will also get made. And then I can just hit my cell or my organism with UV light and watch it glow and be like, oh, my gene just got turned on. I can also get it where it binds to the protein and it will now migrate to wherever the protein's going. So I can be like, oh, it's not even just turned on. I know it's going to this part of the cellular machinery now and stuff like that. In fact, this technology has been around so long that you can go to Petco because it's America and you can buy fish that they've done this to that glow. And so what they've done is they've moved this protein from Aqua Victoria jellyfish into things. And I think you can buy rabbits now. I don't know. Things are going to get weird, folks. That's just the way the whole world is, right? An interesting one is when you start looking at plants. So plants, for the most part, will have fluorescence, like I talked about before, in the red spectrum due to chlorophyll A. But some plants will have this really wild spectrum, but only on the cotyledons. So some of the cotyledons will shift into these radically different colors than red. So we have like a non-native milk thistle in California that probably the easiest way to go and do remediation of this on a trail is go out at night with a UV torch and just pull everything that's blue that's showing up. Tips for the UV lights. There's been a lot of them that have come out over the last 10 years. And I started out with one of these ones, a pet urine light, because it was the only thing that was available for cheap. I mean, geologists have had lights for a long time, but I wasn't willing to get like a generator that straps onto your back so I can have like a giant, UV tube light walking around. But these pet urine lights came out maybe about 10 years ago and they're running about $20. Sounds like a decent price point. The cool thing about them is it gave you access really quickly. The only bad thing was it's not a very strong wavelength. So it doesn't really light up as much as the contemporary lights that just came out, which are these 365 nanometer lights. The best thing you can get right now is a 365 nanometer filtered light that's a C8 style. If you type in C8, like something like Amazon, you'll show the flagship one, which is like $80. But you can buy all these knockoff ones that should be about $30. If you already have one of the pet urine lights that produces the blue and purple spectrum, which makes it hard to see the fluorescence, what you can do is you can get a pair of range glasses or like yellow glasses, and that will get rid of the blue and purple spectrum, and you'll just see the fluorescence. But if you can get these filtered ones, you don't need the lights. Just everything will have much, much more fluorescence. And then this is still in a wavelength that's safe to you. So this is not a wavelength that's going to damage your biological molecules. When you get beneath 300 nanometers, then you need to start wearing protective gear so that you're not just cross-linking your DNA, or screwing up your proteins. So 365, if you can get it, is the way to go if you're just getting started. When you start to go look at plants, you'll see some interesting things just to be aware of. I'm giving this context just once again because I think it's important to understand kind of the broad amount of things you might see in the forest and to get you thinking about the biology behind that. So a lot of white flowers will have red fluorescence. And remember, chlorophyll A is what causes that red fluorescence. What are petals? They're modified leaves. And so what we're seeing is we're seeing the hallmark of a leaf. We're seeing some chlorophyll still in there. So a lot of white flowers will have really strong red fluorescence. One thing to be aware of, though, because this is something that unfortunately, even like I think National Geographic sort of propagated to some degree, when we see UV fluorescence, this is not what bees see. Like if bees saw this, bees would basically just be like tripping acid all the time. What bees actually see is what's on the right-hand side here, which is the UV spectrum. So when we see UV fluorescence, what we're seeing is all those compounds that can absorb UV and then reflect it back out at a different wavelength. What bees are seeing is they can actually see what's absorbing the UV. And so that's why your sunflower, right, becomes this kind of target shape and not this speckled patterns of light. And so these are photographs provided by Michelle Wong from Texas, who is one of the coolest characters if you ever meet her. I think she's like a retired woman. If you meet her, she shows up with like 3 DSLR cameras, one for white light, one for UV fluorescence, and then one that she's made a modified CCD chip in that can see the UV spectrum. So she'll basically shoot everything for her INAT using all 3 spectrums, which gives us a really, really good vision into what's going on. And she's starting to do this with mushrooms too, because she's also a fungophile. So those should be some really, really cool data to share out of hers going forward.
What's her INAT name?
It might just be Michelle Wong. I'll have to look. I tag her in the backside of this deck so we can check. But a lot of these flowers will have really cool fluorescence, a lot of them leaning into the red spectrum, but we won't get further into that. A lot of pollen, though, will be the thing that fluoresces. And pollen is a really interesting thing because it will fluoresce under an enormous wavelength of light. Everything from like the low UV all the way up into like the blue spectrum will cause different types of fluorescence. And this is why a lot of like CSI crime labs like to use, well, not in the US because we're getting worse and worse at science, but a lot of CSI crime labs in Europe use pollen as like a very good tool to understand where somebody was, because you can identify the pollen really easily due to UV fluorescence. And then based on the size and shape, you can say, it came from this plant and I know where the perp was or where the crime happened, which is pretty cool. That's more or less like other plants. So what I'll do is I'll just kind of call out the people here that have really helped with this deck. It allowed me to use photographs. So Michelle Wong is Puffer Chung on INAT. Jim Park gave me some of the stuff to use. Paul helped out with all the millipedes. Morgan was a lot of the plants. Cricket got me the bird stuff, and then Alan provided a lot of the mycena. So I just want to say thank you to them for providing a lot of the images for this. And then one thing we didn't go into is that a lot of guttation also fluoresces. So if you're seeing Fomatopsis, you're seeing Ganoderma, and it's like full of drippy stuff, throw the UV light on there and you'll see fluorescence in that. And that's mainly because a lot of those alkaloids, right, are kind of getting pushed out into the guttation. So if anybody wants this deck, you can kind of get it from that little QR code at the top left. And if you want my contact information, I'm right down there at the bottom right. And we can dig into some of the things that came through in the Q&A.
Thank you, Damon. That was awesome. If anybody does have any questions, go ahead and drop them in the Q&A because I think Allison Pollock had asked briefly about slime molds and them fluorescing, which you touched on briefly. Someone else pointed out how lichens fluoresce. Brian Hunt has some interesting information in a comment here that I'm going to go ahead and read while you guys throw some questions at us. He says, light info about fungi. A few mushrooms have the appearance of being fluorescent when shining them with 365 nanometer ultraviolet light, even though they are not, as light passes through air and into another semi-clear material, such as translucent mushrooms. It changes speed, and light is both reflected and refracted by the mushrooms, giving them a cool look. The first 2 pics that he dropped in the comment show an example of the light differences in Phycomyces species found growing from raccoon scat. So does that mean that the Phycomyces that you shared, that's the same thing, or some of them are actually UV
It,
fluorescent.
um, I mean, because I'm seeing the wavelength come back at a different spectrum, I think there is fluorescence going on. Um, but what he is stating, I think could also be correct there, is that sometimes physical structures will then reorient what light you see. So like we see this in butterfly wings. So like butterfly wings don't have pigment. They have microscales, then change how light is reflected. But in that case, you're dealing with all of the white light. So you're getting reflected back just certain spectrums of the light. With a UV torch, if you're only putting 365 nanometers at it, if you're seeing a different color come back, because I don't see in UV, my assumption would be that that is UV fluorescence that you're seeing. And for the phycomyces, I think what we're seeing is it's probably fluorescence in the like some sort of like metabolite of the, that's wrapping around the turd, and that's why we don't see it in the sporophyte. But happy to be wrong though too. So I mean,
Sego wants to know if you can put the last slide up with the QR codes,
oh yeah, let me
if you
do
can
that
put that back
again.
up. Donna Kirk wants to know if you would share your email, and
Yeah,
also wonders about experience with amanitas. I can say that there are some lovely glowing UV fluorescent amanitas. I've never seen a bioluminescent amanita.
yeah. I've not seen a bioluminescent amanita, but most of the amanitas will have like a pale green fluorescence on the gills.
Aaron wants to know if there are any dried fruiting bodies that UV fluoresce. They do. Uh,
Mm-hmm.
I love going through, I have lots and lots of dried fungal collections. And when I get them all out, I love going through and I know that there's some, and even, before they're dry, if you're at a 4A, get them to turn off the lights in the evening and go over the 4A table with everything with your UV light. I know that there's some, they're starting to consider it pretty heavily for identifying Cordonarius.
Mm-hmm.
Um, the UV fluorescence. I know Russula, it's something that we think about with Russula too, although you were saying that they are all more or less the same.
Yeah, they seem to be very similar. One thing to note about dried specimens versus fresh specimens is that the spectrum that you see usually shifts as you dry them. So the fluorescence that you see on the fresh fruiting body is usually very different than what you see on the dried specimen. What I've noticed is a lot of the dried specimens will fluoresce more than fresh specimens. My assumption is because you're concentrating the compounds that are doing the fluorescence, so they kind of hit the concentration level where you can then pick it up with your eye.
I've also seen the opposite where they will glow fresh but not dried.
Oh, interesting. Have I seen
Usually,
that?
I feel like
What do you think?
it's, well, I'm just going by my memory, which isn't great, but I feel like it's been white things that
I
will
was gonna say
reflect, that will glow fresh.
I think I've seen that too with Hygrophorus and some of the Ruslas. Like they lose the
Hmm.
intensity.
Hmm. Scott wants to know about experimenting with UV fluorescence on very old herbarium specimens.
Um, like we just were talking about, usually you will see fluorescence with them, but it's not going to be the same thing as fresh. I was at Q a couple years ago and they let me hit stuff with some UV torches. So I got to see some like 100-year-old specimens and they were still throwing UV fluorescence. Or the really wild thing was the fungal dyes they had that were like, I think over 100 years old on yarn still had UV fluorescence, which I was kind of blown away by. So any of you that are dyers, know that a lot of the species you dye with, like dyer's polypore, that is strongly UV fluorescent, even on the fiber afterwards, which is super
And on your
cool.
hands where you
Yeah.
touch the fiber, and on anything that the dye was in, and your counter all around where you were working.
Yep.
Donna Kirk, we're talking about them drying and the fluorescence. I don't think any fungi maintain their bioluminescence when dried.
Oh, and freeze and Brian said freeze dried here, and I don't know. I haven't tried any freeze dried specimens. That's a good question. But yeah, bioluminescence you won't see because you need the organism's enzymes to be working. And if you've dried them, usually most enzymes are completely shut down at that point.
Donna Kirk wants to know how many lumens of a light do you recommend? She has 1,000 lumen 365 and a 500 lumen 365 and a
I mean,
500 lumen 395.
basically, the higher lumens you can go for, the more that you're going to be able to pick up on a walk. So I would just take your strongest lumens out that doesn't have purple and blue spectrum being emitted, and that's going to give you your best mix to see things that fluoresce. And that's why that C8 style is a really important kind of differentiators because it has a filter on there that's going to get rid of the blue and purple spectrum that come from a lot of the UV producing LED diode or the diodes.
I have the UV Beast, which does that too, I believe, and
Yep.
I really like it. I have some smaller ones when I'm working in smaller quarters or on at the table in a 4A room or something, but taking people on a UV light walk, the UV Beast is great because I can hit things from a distance and sort of everybody can see whether they have one or not.
Yeah, and then UV Beast and a couple other ones allow you to put an extender tube on them too. So that you can throw another battery on there so you can just get even like, higher ramp up of lumens coming out.
All right, lots of people thanking you. They love the talk, lots of interesting information, wonderful as always. Brian says a lot of my 2025 UV observations on INAT
Oh,
Hills 47448 show reactions after freeze drying. That's amazing.
I will be taking a look at those. Thank you, Brian.
That's super, super interesting. All right. Unless anyone else has any last minute questions, I think we're going to wrap it up. We're right about that time. The presentation has been recorded. I believe it will be up on the NAMA website in the near future. That is not my jurisdiction, but uh, that's what we record them for. Thank you, everybody, for showing up. I think we had 133 people at one point
Oh,
tonight,
wow.
so lots of people tuning in to learn about some UV fluorescence and biofluorescence. Thank you so
Yeah.
much, Damon. This was great.
Thank you. This was really fun.
Have a good night.
You too. Bye.